Earth once contained very simple forms of life that were quite different from modern organisms. During the Ediacaran period, the first large and complex multicellular organisms appeared in the oceans. Most of these life forms had soft bodies and lived on or near the seafloor. After this, the Paleozoic Era marked a major period of biological development. Many organisms evolved hard shells, improved sensory structures, and the ability to move. Marine ecosystems became more diverse, and life gradually expanded into new environments, including land. This period established many of the basic features seen in later plants and animals. At the beginning of Earth's history, conditions were very different from those of today. The planet formed about 4.5 billion years ago, during a time when the solar system itself was still taking shape. The young Earth was extremely hot, with a surface covered in molten rock. Volcanoes were active across the planet, releasing large amounts of gases into the atmosphere. These gases included water vapor, carbon dioxide, methane, and ammonia. There was no oxygen in the air at this time, which is very different from the oxygen-rich atmosphere that supports life today. As the planet slowly cooled, the water vapor in the atmosphere began to condense, forming clouds and eventually rain. This rain fell over long periods of time, filling low areas on the surface and creating the first oceans. These early oceans played a crucial role in the origin of life. They provided a stable environment where chemical reactions could take place. Within these waters, simple molecules began to interact with one another in new and increasingly complex ways. Scientists believe that life began through a gradual process known as chemical evolution. This process did not happen suddenly or in a single moment. Instead, it took place over millions of years as simple chemicals combined to form more complex structures. These structures eventually led to the formation of molecules that could copy themselves. The ability to make copies is one of the most important features of life. Without it, life could not continue or evolve. One of the key ideas in understanding the origin of life is that simple organic molecules formed naturally under early Earth conditions. Experiments carried out by scientists have shown that when gases similar to those of the early atmosphere are exposed to energy sources such as lightning, they can produce amino acids. Amino acids are the building blocks of proteins, which are essential for all living organisms. This suggests that the basic components of life could have formed without the need for existing life. Over time, these small molecules joined together to form larger and more complex compounds. Some of these compounds may have formed structures similar to modern cells. These early cell-like structures were not alive in the way we understand life today, but they represented an important step toward true living organisms. They could separate their internal environment from the outside world, which allowed chemical reactions to take place more efficiently. The first true living organisms were likely very simple. They were single-celled and did not have a nucleus or other complex structures. These organisms are often compared to modern bacteria. They lived in environments that would be considered extreme by today's standards, such as deep-sea hydrothermal vents or hot mineral-rich pools. These environments provided the energy and chemical materials needed for life to survive and grow. Early life forms did not rely on oxygen since there was none available. Instead, they used other chemical processes to produce energy. This type of metabolism is known as anaerobic metabolism. It allowed early organisms to survive in conditions that would be deadly to most modern life forms. Over time, some organisms developed new ways of obtaining energy, including the ability to use sunlight through a process known as photosynthesis. The development of photosynthesis was a major turning point in the history of life on Earth. Certain microorganisms began to use sunlight to convert carbon dioxide and water into energy, releasing oxygen as a byproduct. At first, this oxygen reacted with other elements and did not accumulate in the atmosphere. However, as more and more photosynthetic organisms appeared, oxygen levels slowly increased. This change had a profound impact on the planet. The rise of oxygen in the atmosphere led to what is known as the Great Oxygenation Event. This event changed the chemistry of the oceans and the atmosphere, making it possible for new types of life to evolve. Oxygen allowed organisms to produce energy more efficiently, which supported the development of more complex forms of life. However, it was also toxic to many of the early anaerobic organisms, leading to their decline or extinction. Studying early life is important for several reasons. First, it helps scientists understand the basic principles of biology. By looking at the simplest forms of life, researchers can learn about the fundamental processes that all living organisms share. These include the ability to grow, reproduce, respond to the environment, and maintain internal balance. Second, the study of early life provides insights into the history of our planet. Fossils of ancient microorganisms, along with chemical signatures preserved in rocks, offer clues about the conditions that existed billions of years ago. These clues help scientists reconstruct the environment of early Earth and understand how it changed over time. Third, this field of study has important implications for the search for life beyond Earth. By understanding how life began here, scientists can better identify places where life might exist elsewhere. For example, they look for planets or moons with liquid water, suitable temperatures, and the right chemical ingredients. The discovery of even simple life beyond Earth would have a huge impact on our understanding of the universe. Another important aspect of studying early life is that it shows how life can adapt to extreme conditions. Many modern microorganisms, known as extremophiles, live in environments that are very hot, very cold, highly acidic, or extremely salty. These organisms provide a glimpse into what early life may have been like. They also expand our understanding of the range of conditions under which life can exist. The origin of life is still not fully understood, and many questions remain unanswered. Scientists continue to explore different ideas and conduct experiments to test their theories. Some researchers study the chemistry of early Earth, while others examine ancient rocks for evidence of early organisms. Advances in technology have made it possible to analyze tiny details that were once impossible to detect. One of the challenges in studying early life is that evidence from such a distant time is often scarce and difficult to interpret. Over billions of years, geological processes such as erosion, heat, and pressure have altered or destroyed many of the original records. Despite this, scientists have found important clues in certain types of rocks that have remained relatively unchanged. These rocks contain fossils and chemical markers that provide valuable information about early life. Another area of research focuses on the possibility that life may have originated more than once or in different locations. Some scientists suggest that life could have begun in multiple environments, such as deep-sea vents, shallow ponds, or even within rocks beneath the surface. Each of these environments offers different conditions that could support the formation of complex molecules. There is also the idea that some of the building blocks of life may have come from space. Meteorites and comets contain organic molecules, and it is possible that these materials were delivered to Earth during its early history. This idea, known as panspermia, suggests that the ingredients for life are widespread in the universe. However, it does not fully explain how life itself began. The earliest organisms laid the foundation for all life that followed. Over time, they evolved into more complex forms, leading to the diversity of life we see today. Every plant, animal, and microorganism is connected to those early beginnings through a long chain of evolution. By studying these origins, we gain a better understanding of our own place in the history of life. The Hadean and Archean eras describe the earliest stages in Earth's history, a time when the planet was forming, cooling, and slowly becoming a place where life could begin. These periods are extremely important because they set the foundation for everything that came later, including the appearance of the first living organisms long before the Ediacaran Period. To understand how life began, it is necessary to explore the conditions of early Earth, the chemical changes that took place, and the gradual emergence of the first simple forms of life. During the Hadean era, which began about 4,500,000,000 years ago, Earth was a very different place from what it is today. The surface was extremely hot, with temperatures high enough to melt rock. The young planet was constantly bombarded by space debris, including asteroids and comets. These impacts were so frequent and so powerful that they repeatedly reshaped the surface. One of the most important events of this time was the massive collision that led to the formation of the Moon, often explained by the giant impact hypothesis. This event likely involved a Mars-sized body crashing into Earth, throwing debris into space that later came together to form the Moon. The atmosphere during the Hadean era was also very different. There was no oxygen available for breathing. Instead, the air was filled with gases such as carbon dioxide, methane, ammonia, and water vapor. These gases came mainly from volcanic eruptions, which were extremely common at the time. Volcanoes released heat and chemicals from deep inside the Earth, shaping both the atmosphere and the surface. There were no oceans at first, but as the planet slowly cooled, water vapor in the atmosphere began to condense. Over time, this led to the formation of the first oceans, covering large parts of the planet's surface. Even though conditions during the Hadean era were harsh, scientists believe that important chemical processes were already taking place. These processes may have led to the formation of simple organic molecules, which are the basic building blocks of life. Energy from lightning, volcanic activity, and ultraviolet radiation from the sun could have driven reactions between the gases in the atmosphere and the chemicals in the oceans. Over time, these reactions may have produced more complex molecules, such as amino acids and nucleotides, which are essential for life. As the Hadean era came to an end and the Archean era began, the Earth became somewhat more stable. The surface cooled further, and solid crust began to form. This crust was not as thick or as stable as modern continents, but it marked an important step in Earth's development. Small land masses may have formed and broken apart repeatedly due to intense geological activity. Beneath the surface, the planet remained very hot, and this heat continued to drive volcanic activity and the movement of tectonic plates. The Archean atmosphere still lacked oxygen, but it began to change slowly over time. Carbon dioxide remained a major component, and methane may have been present in significant amounts. These gases helped trap heat, keeping the planet warm even though the sun was less bright than it is today. This is sometimes referred to as the faint young sun paradox, where the early sun provided less energy, yet Earth remained warm enough to support liquid water. The oceans that formed during this time played a key role in the development of life. They provided a stable environment where chemical reactions could occur more easily. In these ancient waters, simple molecules could combine and interact over long periods. Some scientists believe that life may have begun near hydrothermal vents on the ocean floor. These vents release hot, mineral-rich water from beneath the Earth's crust, creating unique environments where chemical energy is abundant. Such conditions could have supported the formation of the first living systems. The earliest signs of life from the Archean era come from structures known as stromatolites. These are layered formations created by communities of microorganisms, particularly a type of bacteria called cyanobacteria. Stromatolites are among the oldest known fossils, with some dating back more than 3,400,000,000 years. They provide strong evidence that life had already emerged and was beginning to shape the environment. Cyanobacteria were especially important because they were capable of photosynthesis, a process that uses sunlight to produce energy. As a byproduct of this process, they released oxygen into the atmosphere. At first, this oxygen reacted with iron and other elements, preventing it from building up in the air. However, over time, oxygen levels began to increase, leading to major changes in Earth's atmosphere and paving the way for more complex forms of life in later eras. In addition to stromatolites, scientists have found other clues about early life in the Archean rocks. These include tiny fossilized cells and chemical signatures that suggest biological activity. For example, certain carbon isotopes found in ancient rocks indicate that living organisms were present and using carbon in ways similar to modern life. These discoveries show that life began relatively early in Earth's history, even under conditions that might seem hostile today. The transition from simple chemistry to living organisms is one of the most fascinating and challenging questions in science. It involves the emergence of systems that can grow, reproduce, and respond to their environment. One important step in this process may have been the formation of self-replicating molecules, such as RNA. RNA can store genetic information and also act as a catalyst for chemical reactions. This has led to the idea of an RNA world, in which RNA played a central role in the earliest forms of life. Over time, these simple systems may have become more complex, leading to the development of cells. The first cells were likely very simple, lacking a nucleus or other specialized structures. These early cells, known as prokaryotes, include bacteria and archaea. Despite their simplicity, they were highly adaptable and capable of surviving in a wide range of environments. The Archean era also saw the gradual development of Earth's crust and continents. small pieces of crust, known as cratons, began to form and stabilize. These cratons are the oldest parts of modern continents and can still be found today in regions such as parts of Africa, Australia, and North America. Their formation marked an important step in the geological evolution of the planet, providing stable areas where life could continue to develop. Their formation marked an important step in the geological evolution of the planet, providing stable areas where life could continue to develop. Another important feature of the Archean era was the interaction between life and the environment. Even the earliest microorganisms had an impact on their surroundings. For example, the oxygen produced by cyanobacteria eventually led to the oxidation of iron in the oceans, forming banded iron formations. These layers of iron-rich rock are found in many parts of the world and provide evidence of the changing chemistry of Earth's oceans and atmosphere. As the Archean era progressed, life continued to evolve and diversify, although it remained microscopic and relatively simple. There were no plants, animals, or complex ecosystems like those seen in later periods. However, the processes that were established during this time laid the groundwork for all future life. The development of photosynthesis, the formation of stable continents, and the gradual increase in oxygen were all crucial steps in Earth's history. By the end of the Archean era, around 2,500 million years ago, the stage was set for the next major phase in Earth's development. Oxygen levels were beginning to rise, leading to what is known as the Great Oxidation Event in the following era. This event would dramatically change the atmosphere and allow for the evolution of more complex forms of life. The Hadean and Archean eras may seem distant and difficult to imagine, but they are essential for understanding the origins of life. During these early times, the Earth transformed from a hot, lifeless world into a planet capable of supporting living organisms. The first signs of life appeared, and the basic systems that sustain life today were established. The Ediacaran Period is one of the most fascinating chapters in the long history of Earth, a time when the planet was changing in deep and powerful ways, and when life began to take on forms that seem almost familiar, yet still strange and mysterious. This period stretched from roughly 635 million years ago to about 540 million years ago, a span of time that followed one of the most extreme and dramatic phases in Earth's past, and led directly into another period of rapid biological change. To understand the Ediacaran period is to step into a world that was both recovering from hardship and quietly preparing for a burst of life that would reshape the surface of the planet forever. At the beginning of the Ediacaran period, Earth had just emerged from a series of severe global glaciations, often described as snowball Earth events. During those earlier times, vast ice sheets had spread across much of the planet, possibly even reaching close to the equator. Oceans may have been covered by thick ice, and life, if it survived at all, had to endure under very harsh conditions. As the Ediacaran began, the planet slowly warmed. Volcanoes released gases such as carbon dioxide into the atmosphere, and over time these gases trapped heat, allowing the ice to melt. The melting of these enormous ice sheets caused dramatic changes. Sea levels rose, fresh water poured into the oceans, and nutrients that had been locked away in rocks and ice began to flow into the seas. The climate of the Ediacaran period was generally warmer than the icy world that came before it, but it was not entirely stable. There were shifts and variations, and different regions of the Earth likely experienced different conditions. However, compared to the frozen ages that preceded it, the Ediacaran was a time of relative warmth and recovery. The atmosphere itself was also changing. Oxygen levels, which had been slowly increasing over hundreds of millions of years due to the activity of simple photosynthetic organisms, reached levels high enough to support more complex life forms. This rise in oxygen was one of the most important factors that made the Ediacaran period unique, because it allowed organisms to grow larger and develop more complex structures. The oceans during the Ediacaran period were central to everything that was happening. Life at this time was almost entirely marine, and the seas became a stage for experimentation in form and structure. The chemistry of the oceans was changing as well. Increased oxygen levels affected how elements such as iron and sulfur behaved in seawater, and this influenced the types of environments that could exist. In some areas, the water was rich in oxygen, while in others, especially deeper regions, it may still have been low in oxygen or even toxic. These differences created a wide range of habitats, each with its own challenges and opportunities for life. The seafloor itself was very different from what we might imagine today. Instead of being filled with burrowing animals and complex ecosystems, much of the seabed was covered with microbial mats. These mats were thick layers of microorganisms, mainly bacteria and other simple life forms, that formed a kind of living carpet. They played an important role in shaping the environment. They could trap sediments, stabilize the surface, and even influence the chemistry of the water just above them. These microbial mats provided both a foundation and a boundary for the new forms of life that began to appear. One of the most remarkable features of the Ediacaran period is the appearance of large, soft-bodied organisms. These life forms, often grouped together as the Ediacaran biota, represent some of the earliest examples of complex multicellular life. They did not have hard shells or bones, which is why they are mostly known from impressions preserved in sedimentary rocks. Their shapes were often unlike anything seen in later periods. Some looked like flat discs, others like fronds or leaves, and still others had elongated, segmented bodies. Many of them were anchored to the seafloor, while others may have rested gently on the microbial mats. These organisms were not only unusual in shape, but also in the way they lived. Unlike many modern animals, they did not seem to have mouths, digestive systems, or obvious organs. Some scientists believe they absorbed nutrients directly from the water through their bodies. Others may have had symbiotic relationships with microorganisms, relying on them for energy. Because their biology is so different from that of later animals, it is still debated exactly how they fit into the tree of life. Some may represent early relatives of modern groups, while others could belong to entirely extinct branches that left no descendants. The environment of the Ediacaran seas was generally calm and stable in many places, especially in shallow marine settings where these organisms thrived. Gentle currents moved water and nutrients across the seafloor, and sunlight penetrated the clear waters, supporting photosynthetic life. However, the stability of these environments could be interrupted by events such as storms, changes in sea level, or shifts in ocean chemistry. When sediments were rapidly deposited, they sometimes covered the soft-bodied organisms, preserving their shapes as fossils. This is one of the reasons why we have such a detailed record of Ediacaran life in certain regions. The continents during the Ediacaran period were arranged very differently from today. Earlier in Earth's history, many of the landmasses had been joined together in a supercontinent known as Rodinia. By the time of the Ediacaran, this supercontinent had begun to break apart. The fragmentation of Rodinia led to the formation of new coastlines and shallow seas, which were ideal environments for life to develop. The movement of tectonic plates also contributed to volcanic activity, which in turn influenced the atmosphere and climate. These geological processes were constantly reshaping the surface of the planet, creating new habitats and altering existing ones. Weather patterns during the Ediacaran period were influenced by the changing arrangement of continents and oceans. With more open ocean areas and different land distributions, currents and wind patterns would have been different from those of today. Rainfall, evaporation, and the cycling of water through the atmosphere all played roles in shaping the environment. Although we cannot observe these processes directly, scientists study rock formations and chemical signatures to reconstruct what conditions might have been like. Another important aspect of the Ediacaran environment was the cycling of nutrients. As glaciers melted and rocks were weathered, nutrients such as phosphorus and nitrogen were released into the oceans. These nutrients supported the growth of microorganisms, which formed the base of the food web. The availability of nutrients likely varied from place to place, leading to differences in productivity and the types of organisms that could thrive. In areas where nutrients were abundant, life may have been more diverse and abundant, while in poorer regions, it may have been more limited. The Ediacaran period also saw changes in the way sediments were deposited and preserved. Because there were few organisms capable of burrowing into the seafloor, sediments often remained undisturbed after being laid down. This allowed delicate structures, including the impressions of soft-bodied organisms, to be preserved with remarkable detail. In later periods, the activity of burrowing animals would disrupt sediments, making such preservation much less common. In this way, the Ediacaran provides a unique window into a style of life and environment that would soon be transformed. As the Ediacaran period progressed, there were signs of increasing biological complexity. Toward the later part of this time, some organisms began to show features that hint at more advanced forms of life. There is evidence of simple movement, such as trails or tracks left behind on the seafloor. These traces suggest that some organisms were capable of moving across the surface rather than remaining fixed in place. This was an important step in the evolution of behavior, as movement allows organisms to seek out food, avoid danger, and interact with their environment in new ways. The end of the Ediacaran period marks a transition into the Cambrian period, a time often associated with a rapid diversification of life known as the Cambrian explosion. While the Ediacaran itself was not as dramatic in terms of the variety of forms, it laid the groundwork for what was to come. The increase in oxygen levels, the development of new body plans, and the creation of more complex ecosystems all set the stage for the burst of evolutionary innovation that followed. Climate changes toward the end of the Ediacaran may have played a role in shaping this transition. Fluctuations in temperature, sea level, and ocean chemistry could have created pressures that favored certain types of organisms over others. Some of the unique Ediacaran forms disappeared, possibly because they could not adapt to the changing conditions or compete with emerging groups. Others may have evolved into new forms that would become more recognizable in the fossil record of the Cambrian. The Ediacaran period is often described as a time of quiet preparation, but this does not mean it was simple or uneventful. On the contrary, it was a period filled with experimentation and change. Life was exploring new possibilities, trying out different shapes, sizes, and ways of living. The environment was dynamic, shaped by forces both internal and external, from the movement of tectonic plates to the composition of the atmosphere. One of the most intriguing aspects of the Ediacaran world is how different it was from our own, yet how it still connects to the present. The basic processes that governed the Earth then, such as the cycling of carbon, the movement of plates, and the flow of energy through ecosystems, are the same processes that operate today. By studying the Ediacaran period, scientists gain insight into how these processes interact and how they can lead to major changes over long periods of time. The fossil record from this period, though limited to certain types of environments, provides valuable clues. Sites around the world have revealed well-preserved impressions of Ediacaran organisms, allowing scientists to study their shapes and distributions. These fossils are often found in fine-grained sedimentary rocks, such as sandstone or siltstone, which captured the delicate forms of the organisms before they decayed. Each discovery adds to our understanding of what life was like during this time and how it evolved. Despite all that has been learned, many questions about the Ediacaran period remain unanswered. The exact relationships between the various organisms are still debated, as is the nature of their biology and ecology. Some researchers suggest that certain Ediacaran forms may represent early animals, while others argue that they belong to entirely separate groups. New techniques, including advanced imaging and chemical analysis, are helping to shed light on these mysteries, but there is still much to discover. The environment of the Ediacaran period was not only shaped by biological and geological processes, but also by chemical interactions within the oceans and atmosphere. The balance of gases, the availability of nutrients, and the movement of water all influenced how life developed. These interactions created a complex system in which changes in one area could have effects in others. For example, an increase in oxygen levels could alter ocean chemistry, which in turn could affect the types of organisms that could survive. The Ediacaran biota refers to a remarkable group of soft-bodied organisms that lived in Earth's oceans during a distant stretch of time, roughly between 635 million and 540 million years ago. These life forms represent some of the earliest large and complex organisms ever known, appearing after billions of years during which life was mostly microscopic. Unlike later animals that developed hard shells, bones, or teeth, these organisms were delicate and lacked rigid parts, which makes their discovery in the fossil record both rare and extraordinary. Their remains were preserved only under special conditions, often as faint impressions in ancient rock layers, giving scientists just enough evidence to begin piecing together what these early forms of life may have looked like and how they lived. The environment in which the Ediacaran biota thrived played a major role in shaping their appearance and way of life. The oceans were rich in nutrients, and oxygen levels were rising compared to earlier times in Earth's history. This increase in oxygen is believed to have supported the growth of larger and more complex organisms. However, even with more oxygen available, the ecosystems were still very simple compared to those that would come later. There were no predators in the modern sense, no animals with jaws or sharp teeth, and no fast-moving hunters chasing prey. Instead, most organisms lived quietly on or near the seafloor, absorbing nutrients directly from the water or from microbial mats that covered the ocean floor like living carpets. These microbial mats were extremely important in the Ediacaran world. They were made up of layers of bacteria and other microscopic life forms that formed thick, sticky surfaces across the seabed. The mats not only provided food, but also helped preserve the shapes of soft-bodied organisms when they died. When an organism settled onto the mat, it could leave a clear impression that later became fossilized as sediments covered it. Without these mats, much of the evidence of Ediacaran life might have been lost forever. One of the most striking features of the Ediacaran biota is the incredible variety of shapes and forms they displayed. Many of these organisms do not resemble any modern animals, which has made them difficult to classify. Some were flat and disc-shaped, lying on the seafloor like round cushions. Others had long, leaf-like bodies with repeating patterns that extended outward in a symmetrical way. These repeating patterns, often described as fractal-like, gave the organisms a sense of balance and order, even though their exact function remains uncertain. Among the most well-known Ediacaran organisms is Dickinsonia, a creature that had an oval shape with rib-like segments running across its body. It could grow to a considerable size, sometimes reaching lengths of over a meter, which was enormous for its time. The body of Dickinsonia appears to have been soft and flexible, and it likely moved slowly across the seafloor, possibly feeding by absorbing nutrients from the microbial mats beneath it. Some fossil evidence even suggests that it may have left behind feeding traces as it moved, indicating a simple form of behavior. Another fascinating organism is Charniodiscus, which had a tall, frond-like structure anchored to the seafloor by a stem. Its body spread out into a leaf-shaped form with a central axis and branching segments on either side. This structure may have helped it capture nutrients directly from the water, much like modern sea pens or certain types of algae. However, unlike plants, these organisms did not perform photosynthesis, and their exact method of feeding remains a subject of study. There were also organisms like Sprigina, which had a more complex appearance, with a head-like region and a series of segments along its body. Some scientists believe that Sprigina may be related to early animals that later gave rise to arthropods, a group that includes insects, spiders, and crustaceans. If this connection is correct, it would mean that the roots of many modern animals can be traced back to these early forms. However, this idea is still debated, as the features of Sprigina are not clearly understood. Another unusual form is Tribrachidium, which had a unique three-part symmetry. Unlike most modern animals, which have bilateral symmetry with left and right sides, or radial symmetry like a starfish, Tribrachidium displayed a three-armed pattern that is rarely seen in nature today. This strange body plan suggests that the Ediacaran world experimented with a wide range of biological designs, many of which did not survive into later periods. The lack of hard body parts in these organisms means that scientists must rely on indirect evidence to understand how they lived. Fossils often show only the outline or impression of the organism, leaving questions about their internal structures unanswered. Did they have organs? Did they have nervous systems? Could they sense their environment? These questions are still being explored. Some researchers suggest that many Ediacaran organisms may have absorbed nutrients directly through their skin, a process known as osmosis, which would have been efficient in the nutrient-rich waters of the time. Movement among the Ediacaran biota was likely very limited. While some organisms, like Dickinsonia, appear to have been capable of slow movement, most were probably fixed in place. Anchored forms like Charniodiscus would have remained attached to the seafloor, relying on water currents to bring nutrients to them. This lifestyle reflects a world where energy demands were low and competition was minimal, allowing simple forms of life to thrive without the need for speed or complex behavior. The absence of predators during much of the Ediacaran period also had a profound impact on the evolution of these organisms. Without the threat of being eaten, there was little need for defensive structures such as shells or spines. This is one reason why so many Ediacaran organisms remained soft-bodied. However, toward the end of the period, there is some evidence that simple forms of predation may have begun to appear, possibly contributing to changes in the ecosystem and the eventual disappearance of many Ediacaran forms. The extinction of the Ediacaran biota is another mystery that continues to intrigue scientists. As the Cambrian period began, a new wave of life forms emerged, many with hard shells and more complex body structures. This period, often called the Cambrian explosion, saw the rapid diversification of animal life. Many Ediacaran organisms seem to have vanished during this transition, possibly due to environmental changes, competition with new species, or the rise of predators. Some may have evolved into later forms, while others disappeared entirely. Despite their disappearance, the Ediacaran biota left a lasting legacy. They represent an important step in the evolution of life, marking the transition from simple, microscopic organisms to larger and more complex forms. They also show that evolution is not always a straight path, but rather a process filled with experimentation, where many different forms arise and only some continue into the future. The study of these organisms has been made possible by fossil discoveries in various parts of the world, including regions in Australia, Russia, Canada, and Namibia. These sites have provided valuable insights into the diversity and distribution of Ediacaran life. Each new discovery adds another piece to the puzzle, helping scientists better understand this ancient world. What makes the Ediacaran biota especially fascinating is how different they are from modern life. Many of their forms have no clear modern equivalents, which challenges our understanding of what life can be. The Ediacaran seas were a place of quiet complexity, where soft-bodied organisms spread across the ocean floor in a variety of shapes and sizes. They did not swim or hunt in the way later animals would, but they formed communities, interacted with their environment, and laid the groundwork for the explosion of life that followed. In their simplicity and strangeness, they offer a glimpse into a time when life was still exploring its possibilities, testing different forms and strategies in a world that was just beginning to support complexity. The Ediacaran period, which stretches from about 635 million years ago to around 540 million years ago, represents one of the most fascinating chapters in the history of life on Earth. During this long and mysterious stretch of time, the seas were filled with life forms unlike anything that exists today, yet they provide the earliest glimpses of complex multicellular life. The creatures that dominated these ancient oceans are collectively known as the Ediacaran biota, and among them, some of the most remarkable and widely studied organisms include Dickinsonia, Charnia, Sprigina, and a variety of other extraordinary species. These organisms are central to our understanding of early animal life because they represent some of the first large, complex creatures on the planet, and their fossils tell us about the beginnings of ecosystems and behaviors that would later shape the evolution of all life forms. One of the most iconic Ediacaran organisms is Dickinsonia, a strange, flat, segmented creature that is immediately recognizable to anyone who studies Ediacaran fossils. Dickinsonia appears as an oval or elongated shape, with a series of repeated segments arranged symmetrically along its central axis. The symmetry is not exactly like the bilateral symmetry seen in modern animals. It is more irregular, with segments often varying slightly in size, giving each individual a unique pattern. Fossils of Dickinsonia are generally preserved as impressions in sandstone, showing the outline of the soft-bodied organism rather than bones or shells, which were absent in these early life forms. What makes Dickinsonia truly fascinating is that scientists have debated its classification for decades. Some researchers once considered it a type of fungus, or even a giant amoeba, while others argued that it represented a very early animal. Recent studies, including chemical analyses of fossilized tissues, have provided compelling evidence that Dickinsonia was indeed an early animal, one of the earliest forms of complex multicellular life capable of movement and feeding across the seafloor. Dickinsonia likely fed by absorbing nutrients from microbial mats, layers of bacteria, and other tiny organisms that blanketed the ancient seafloor, leaving behind telltale traces of its movement as it glided over these mats. Another remarkable organism from the Ediacaran seas is Charnia, a frond-like creature that looks unlike any modern animal. Charnia fossils are characterized by their branching, leaf-like structures that rise from a central stalk. They were attached to the seafloor, probably anchored in the soft sediments, and are believed to have been stationary filter feeders, absorbing nutrients directly from the water. The discovery of Charnia was revolutionary because it was one of the first Ediacaran fossils to be formally described, opening the door to the recognition of a whole new group of ancient organisms. Unlike Dickinsonia, which could move, Charnia remained fixed in place, and its shape suggests a life that was adapted to capturing whatever food particles drifted by in the currents. Its delicate fern-like structure might seem fragile, but it was a highly successful design for surviving in the calm, shallow seas of the late Precambrian. Charnia's fossils are often preserved as carbon films in sandstone, highlighting the intricate patterns of its fronds and offering a window into the soft-bodied diversity of Ediacaran life. Sphrygina is another Ediacaran organism that has intrigued paleontologists for decades. Sphrygina fossils display a segmented body with a clearly defined head region and a tapering tail, giving it a more recognizable animal-like form compared to other Ediacaran creatures. Its head often features a small, crescent-shaped structure, while the rest of its body is composed of a series of repeating segments. These features led scientists to speculate that Sphrygina might represent an early ancestor of arthropods or annelid worms, though its exact relationships remain uncertain. Evidence from fossil beds suggests that Sphrygina could move across the seafloor, leaving faint trace fossils behind. Its mobility, combined with its segmented body, makes Sphrygina one of the earliest examples of an organism capable of exploring its environment in search of food, a behavior that would become increasingly important in the Cambrian period that followed. Fossils of Sphrygina are found in well-preserved sandstone layers, often showing impressions of both the body and the traces of movement, providing a rare glimpse into the behavior of Ediacaran animals. Beyond these three famous genera, the Ediacaran seas were home to a remarkable array of other organisms that challenge our understanding of early life. For example, Charniodiscus resembled Charnia in its frond-like appearance, but had a more prominent stalk and a circular holdfast, which anchored it securely in the sediment. Its fronds were arranged in a way that maximized surface area, suggesting an adaptation to capture nutrients efficiently from the water. Another fascinating organism is Sphrygina's relative, Dickinsonidae, which includes a variety of segmented, flat-bodied creatures with slightly different shapes and sizes. These species illustrate the diversity within the Ediacaran biota, showing that even during this early period, life was experimenting with different forms, sizes, and strategies for survival. Some Ediacaran species, like Tribrachidium, display radial symmetry, meaning their body plan radiated from a central point rather than along a line, which is a form of organization rarely seen in modern animals, but was relatively common in the Precambrian seas. Tribrachidium's threefold symmetry and circular shape suggest it may have been a sessile filter feeder, anchored to the seafloor while capturing food from all directions. The preservation of these fossils also tells a story about the environments in which they lived. Most Ediacaran fossils are found in fine-grained sandstone, siltstone, or shale, indicating that these organisms thrived in shallow marine environments where fine sediments settled slowly on the seafloor. Fossilized impressions reveal that many of these creatures were soft-bodied and lacked skeletons, shells, or hard parts, which makes their preservation in the fossil record particularly rare and valuable. The presence of microbial mats played a crucial role in this preservation. These mats covered the seafloor with sticky, layered colonies of bacteria and tiny organisms, which not only served as a food source for creatures like Dickinsonia, but also helped protect and preserve their bodies after death. When an Ediacaran organism settled on a microbial mat, it could leave a detailed impression that, over millions of years, became fossilized in the surrounding sediment. This unique combination of soft-bodied life and microbial mats resulted in fossil beds that provide an extraordinarily detailed record of life in the late Precambrian. The Ediacaran biota also demonstrate remarkable variations in size, shape, and complexity. Some organisms were only a few millimeters long, while others, like Dickinsonia, could grow to over 1 meter in length, showing that life was already experimenting with large body sizes long before the explosion of animal life in the Cambrian period. The diversity of body plans, from flat, oval forms to frond-like structures and segmented creatures, reflects a period of evolutionary experimentation in which early multicellular organisms explored new ways of surviving and thriving in marine environments. Many of these forms do not have direct descendants among modern animals, which has led scientists to classify them as a distinct branch of life, sometimes referred to as the extinct Ediacaran fauna. Nevertheless, some features, such as segmentation and bilateral symmetry, hint at connections to later animal lineages and suggest that the Ediacaran organisms laid the groundwork for the evolutionary developments that would follow in the Cambrian explosion. Understanding these organisms also provides insights into how early ecosystems functioned. In contrast to modern ecosystems, which often rely on predation and complex food chains, the Ediacaran ecosystems appear to have been dominated by organisms that absorbed nutrients from microbial mats or directly from the water. There is little evidence of active predation, though some Ediacaran fossils do show scratch marks and traces that may indicate early attempts at movement for feeding purposes. The interactions between these creatures, such as the overlapping of fronds or the displacement of microbial mats, suggest that competition for space and food was already shaping the distribution and abundance of life on the seafloor. This dynamic, though simple compared to modern marine ecosystems, represents an important step in the development of ecological complexity, demonstrating that even hundreds of millions of years ago, life was engaging in interactions that would become central to later evolutionary history. The discovery of these fossils in multiple locations around the world, including Australia, Russia, Canada, and Namibia, demonstrates that the Ediacaran biota were globally distributed and able to thrive in a variety of shallow marine environments. Sites such as the Ediacara Hills in Australia, from which the period gets its name, provide some of the most famous and well-preserved fossil beds. In these locations, large assemblages of fossils have been discovered together, showing communities of Ediacaran organisms living side by side, often in remarkable abundance. These fossil assemblages reveal patterns of growth, population density, and species diversity that help scientists reconstruct the ancient seafloor and understand the ecological principles that governed life in the late Precambrian oceans. The study of Ediacaran organisms like Dickinsonia, Charnia, and Spergina has revolutionized our understanding of early animal evolution. By examining their body plans, methods of feeding, movement, and interactions with microbial mats, scientists have been able to piece together a picture of a world that is both alien and familiar. These creatures represent the first steps toward the complex life forms that would dominate the planet in the Cambrian period and beyond. They also challenge assumptions about what early animals looked like, how they lived, and how ecosystems functioned hundreds of millions of years ago. Despite their simplicity in some ways, these organisms exhibit a remarkable variety of forms, showing that life was already experimenting with diversity, size, and ecological strategies long before the advent of hard-bodied skeletons and complex predatory relationships. Many Ediacaran organisms were simple in terms of their internal structures, but highly specialized in their external forms. They were soft-bodied, meaning they lacked bones, shells, or hard skeletons, which makes their preservation in fossils remarkable because it required special conditions such as fine sediment and gentle burial that could capture their delicate outlines. The shapes of these organisms were diverse and often symmetrical, a feature that might have helped them maximize exposure to light, water, or nutrients, depending on their mode of life. Some had radial symmetry, like the spokes of a wheel, which allowed them to interact with the environment equally from all sides, while others exhibited bilateral symmetry, which later became a fundamental body plan in most animals, facilitating directional movement and specialized organs. Among the most iconic Ediacaran organisms is Dickinsonia, a flat, segmented organism that resembled a leaf or an oval-shaped mattress. Dickinsonia's body was composed of repeated units arranged along a central axis, a feature called modular construction, where similar body units repeated in sequence. This repetition may have helped Dickinsonia grow by adding more segments as it aged, a strategy that allowed it to reach remarkable sizes, with some specimens growing to more than 4 feet in length. The overall symmetry and segmentation of Dickinsonia suggests that it could have absorbed nutrients from the seafloor, possibly feeding on microbial mats that covered the ocean floor. Its soft, flattened body and broad surface area were likely adaptations that allowed it to efficiently gather nutrients while staying anchored in one place. Another remarkable organism is Charnia, a frond-like creature with a long, tapering body that branched repeatedly into smaller units, creating a feather-like appearance. Unlike modern plants, Charnia did not have roots, leaves, or photosynthetic tissues. Its frond-like structure likely functioned to maximize surface area for feeding, possibly by absorbing dissolved organic matter from the water. Its body grew from the base, which remained attached to the seafloor, while the upper fronds reached upward into the water column. This method of growth is quite different from the way most animals today grow, highlighting the experimental nature of body plans during the Ediacaran period. Charnia's repeated branching pattern demonstrates an early example of fractal-like design in nature, a mathematical strategy that allows a structure to expand efficiently while maintaining strength and stability. Sphrygina is another fascinating Ediacaran organism, smaller and more mobile than Dickinsonia and Charnia, with a bilaterally symmetrical body divided into segments. Its head region was distinct from its body, suggesting that it may have had a sense of direction, moving purposefully across the seafloor in search of food. Sphrygina's segmented body plan hints at early experimentation with forms that later became common in the animal kingdom, such as arthropods and annelid worms. Its morphology indicates it could have been capable of limited movement, making it one of the first examples of motile Ediacaran life. The differentiation of a head and body along with repeated segments may represent an evolutionary step toward more complex animals that can actively seek food rather than passively absorb it from their environment. Several other Ediacaran organisms demonstrated even stranger and more experimental body plans. For instance, Tribrachidium had a unique tri-radial symmetry, with its body divided into 3 repeating sections arranged around a central point. This arrangement is unlike any modern animal and demonstrates that the Ediacaran period was a time of radical experimentation with symmetry and form. Tribrachidium's shape may have helped it stabilize on the seafloor and interact with water currents in a way that allowed it to filter nutrients from multiple directions. Similarly, organisms like Kimberella exhibited a more mollusk-like body with a broad, soft foot and a flexible body that may have allowed it to glide across the seafloor. These varied morphologies highlight that the Ediacaran period was not merely a time of primitive life, but a period of sophisticated biological experimentation, where multiple strategies for survival, feeding, and growth were being tested in soft-bodied forms. The body surfaces of many Ediacaran organisms were also remarkable in their textures and patterns. Some, like Dickinsonia, displayed fine ridges along the length of their bodies, which may have been structural adaptations for flexibility and stability. Others, like Charnia, had smooth, branching fronds that maximized their exposure to water currents, suggesting they relied on passive feeding strategies. Many Ediacaran organisms were relatively flat and close to the seafloor, allowing them to efficiently interact with the microbial mats that covered much of the ocean bottom. These microbial mats were composed of bacteria and other simple life forms, and they provided both food and a habitat for Ediacaran animals. The intimate relationship between the morphology of Ediacaran organisms and the microbial mats illustrates how body plans were shaped by environmental pressures and resource availability, demonstrating an early form of ecological adaptation. Some Ediacaran organisms exhibited radial symmetry with central openings or depressions, suggesting they might have had simple digestive or feeding structures. For example, the disc-shaped form of organisms like Aspidella may have anchored them to the seafloor while allowing them to capture food particles from the surrounding water. The presence of such openings indicates that even though these organisms lacked complex internal organs, they were already developing specialized features for feeding and maintaining their bodies. The diversity of shapes, from flat ovals to branching fronds and tri-radial discs, shows that the Ediacaran period was a time when nature was experimenting with every possible variation of soft-bodied design, unconstrained by the later dominance of skeletons and shells. Growth patterns in Ediacaran organisms were also highly varied. Many, such as Dickinsonia, grew by adding new segments at one end, a process known as terminal addition. Others, like Charnia, grew by expanding existing branches while adding new ones at the base, a method called basal growth. These different strategies suggest that the Ediacaran organisms were exploring multiple ways to increase size and complexity. Some organisms could reach impressive dimensions. For instance, fossil records indicate that Dickinsonia could grow up to more than 4 feet in length, and Charnia fronds could reach over 6 feet, towering above the microbial mats and creating microhabitats for other smaller organisms. These large sizes demonstrate that even in a world dominated by soft-bodied life, organisms could achieve ecological dominance through clever use of their body plans and growth strategies. Another striking aspect of Ediacaran morphology was the development of modular and repeating patterns. Modular construction, as seen in Dickinsonia and Spraguina, involved repeating units that allowed organisms to grow efficiently and maintain flexibility. This repetition might have provided resilience, as damaged segments could be regenerated or new segments could compensate for lost parts. Repetition also allowed the organisms to expand their feeding surface, enhancing their ability to absorb nutrients or capture particles from the environment. The presence of modularity in multiple Ediacaran taxa suggests that this was a successful evolutionary strategy that paved the way for the segmented body plans seen in many later animals, including worms, arthropods, and vertebrates. The experimental nature of Ediacaran body plans extended to their ecological strategies. Some organisms were sessile, anchored to the seafloor, relying on passive feeding from surrounding water or microbial mats. Others were motile, able to glide or crawl in search of food, like Spraguina and Kimberella. Some may have been capable of limited digestion or absorption of organic material directly through their body surfaces, while others had specialized structures for capturing suspended particles. These variations in body plans and feeding strategies illustrate the diversity of life during the Ediacaran and highlight that the period was a crucial stage in the evolution of complex life. Organisms were experimenting not only with shapes, but also with how to survive and exploit the environment in multiple ways. Fossil evidence of Ediacaran organisms also reveals the presence of complex interactions among species. The arrangement of fossils sometimes shows overlapping and interwoven forms, suggesting competition for space, resources, or light. The morphology of these organisms, from broad, flat surfaces to tall fronds, may have been influenced by the need to compete effectively in dense microbial mat communities. Such interactions hint at the early emergence of ecological dynamics that would later become central to the Cambrian explosion, where animal life diversified rapidly and developed more complex forms and behaviors. The remarkable diversity of Ediacaran body plans challenges our understanding of early animal evolution. Many of these organisms do not fit neatly into modern groups, leading scientists to propose that they represent extinct lineages or experimental body forms that left no descendants. The study of their morphology helps paleontologists identify patterns of symmetry, segmentation, modularity, and growth that reveal how life was exploring all possible ways to build a body. This period shows that evolution is not linear, but involves numerous experiments, some of which succeed and become dominant, while others, like many Ediacaran forms, vanish without leaving direct descendants. The ecological roles of these early life forms reveal a remarkable diversity of strategies for living, feeding, and interacting with their surroundings, demonstrating that even before the explosion of life in the Cambrian, the seas were dynamic, teeming, and full of ecological complexity. The organisms of the Ediacaran seas were predominantly soft-bodied and fragile, leaving behind fossils that are often mere impressions on sedimentary rocks. Despite their delicate structures, these creatures occupied a variety of ecological niches, each adapted to the challenges and opportunities of the shallow marine environment. They ranged in size from a few millimeters to more than a meter in length and exhibited an array of shapes and body plans, from flat and frond-like forms to oval, segmented, and quilted designs. These shapes were not merely artistic expressions of nature, they were functional adaptations that allowed the organisms to feed, anchor themselves, reproduce, and interact with other organisms in specific ways. One of the most striking features of Ediacaran life was its relationship with the seafloor. Many of the organisms were sessile, meaning they remained anchored in one place, often on microbial mats that carpeted the shallow seabed. These mats, composed of dense colonies of microorganisms such as bacteria and early algae, provided a vital resource. Sessile Ediacaran organisms, such as the iconic frond-like Charnia, extended their bodies upward from these mats, increasing their exposure to water currents. This positioning likely helped them capture microscopic particles and nutrients drifting in the water. Their fronds could sway with gentle currents, maximizing the surface area exposed to nutrient-rich water and allowing passive feeding through absorption or suspension capture, a method in which tiny organic particles are trapped and taken into the organism's body. This passive feeding strategy highlights the deep interconnection between these early animals and the microbial ecosystems beneath them, as the microbial mats not only stabilized the sediment but also supplied a constant stream of nutrients. Other Ediacaran organisms exhibited movement, albeit limited and slow. For instance, Dickinsonia, a flat, segmented, oval-shaped organism, is thought to have glided across the microbial mats. It likely fed by absorbing nutrients directly from the mats, secreting enzymes that broke down the microbial cells into forms it could ingest. This form of feeding, known as osmotrophy, depended entirely on the organism's close interaction with its environment, showing that even early animals were part of a complex network of energy transfer. Dickinsonia and similar forms may have left subtle traces on the sediment, providing evidence of their slow locomotion and grazing patterns, which are among the earliest signs of active interaction with a living substrate. These interactions also suggest that Ediacaran organisms could have influenced the distribution and composition of microbial communities, much like modern grazers, influence algae and bacteria on contemporary seafloors. Predation, a central ecological interaction in modern ecosystems, was rare or perhaps absent in the Ediacaran. The fossil record shows few signs of biting, piercing, or hunting behaviors. Instead, these early ecosystems were largely built on passive feeding strategies, recycling nutrients within microbial mats and absorbing organic matter directly from the environment. While this may seem simplistic compared to later ecosystems dominated by predatory interactions, it represents an important stage in ecological evolution. The absence of active predation allowed for the development of soft-bodied morphologies, as there was less selective pressure for hard shells, spines, or other defensive structures. This soft-bodied life set the stage for the later Cambrian explosion, when the evolution of predators would drive the rapid diversification of body forms, strategies, and behaviors. Reproduction was another critical aspect of Ediacaran ecological roles. Although the fossil record provides limited direct evidence of reproductive strategies, scientists infer that many Ediacaran organisms reproduced both sexually and asexually. Frond-like organisms may have reproduced by budding, creating clones that spread across the seafloor, while more complex forms like Dickinsonia may have employed sexual reproduction, ensuring genetic diversity. These reproductive strategies influence population structure, distribution, and interactions among species. Asexual reproduction allowed rapid colonization of available space, particularly in environments dominated by stable microbial mats, while sexual reproduction introduced variation that could enhance adaptability to environmental changes. The spatial organization of Ediacaran communities also reflects ecological roles. Fossil beds reveal dense aggregations of organisms arranged in patterns that suggest competition for space and resources. Some areas show high densities of frond-like forms standing upright, while other areas feature scattered, creeping organisms like Dickinsonia. The arrangement of these organisms may have minimized direct competition by exploiting different layers or zones of the microbial mat. For instance, tall fronds accessed nutrients in water currents above the mat, while flat, creeping organisms absorbed nutrients at the mat's surface. This vertical and horizontal partitioning of resources demonstrates a level of ecological complexity that challenges earlier notions of the Ediacaran seas as simple or static environments. The Ediacaran organisms also played an important role in sediment dynamics. By living on and interacting with microbial mats, they contributed to the stabilization or disturbance of sediments. Creeping and feeding organisms may have stirred surface layers, aiding in the redistribution of nutrients, while stationary fronds could have shielded underlying sediments from erosion. These interactions between organisms and their environment reflect early forms of ecosystem engineering, where life actively shapes the physical characteristics of habitats, influencing both biological and geological processes. Another fascinating aspect of Ediacaran ecology was the diversity of body symmetries and forms, which reflect ecological strategies. Many organisms displayed radial symmetry, such as the disc-shaped forms like Aspidella, allowing them to feed in all directions or maintain stability on soft substrates. Others exhibited bilateral symmetry, such as Sprigina, which may have enabled more directional movement and more efficient exploration of their environment. These body plans were not only morphological features, but also functional adaptations, allowing organisms to occupy distinct niches and maximize their access to nutrients. The presence of different symmetries highlights the experimental nature of early multicellular evolution, as organisms explored a wide range of forms and lifestyles in response to environmental pressures. Ediacaran organisms also show evidence of interactions that go beyond simple feeding and movement. Some fossils display patterns that suggest coordinated or collective behaviors. For instance, clusters of frond-like organisms sometimes align in parallel rows, possibly reflecting responses to water currents, light availability, or chemical gradients. These arrangements may have enhanced nutrient uptake, reproduction, or protection from sedimentation. Although we cannot directly observe behavior in extinct organisms, the fossil patterns provide indirect but compelling evidence that Ediacaran life involved more than isolated individuals. These communities were interconnected networks of organisms influencing and responding to one another and their surroundings. Microbial mats, which dominated the Ediacaran seafloor, were central to the ecology of these early ecosystems. These mats acted as both habitat and food source, forming complex surfaces on which Ediacaran organisms could attach, move, or feed. The mats may have consisted of layers of cyanobacteria, green algae, and other microorganisms that produced organic matter through photosynthesis or chemosynthesis. The relationship between Ediacaran organisms and microbial mats was mutual. The mats provided nutrients and support, while the organisms influenced the mat's distribution, composition, and nutrient cycling. In this way, the first complex ecosystems were already demonstrating key ecological principles that persist today, including energy flow, nutrient recycling, and habitat engineering. The ecological roles of Ediacaran organisms also varied with environmental conditions. Fossils show that some species preferred quiet, shallow waters where microbial mats thrived, while others may have tolerated slightly deeper or more turbulent conditions. This environmental sensitivity indicates that these organisms were adapted to specific niches and that ecological diversity was linked to variations in water depth, sediment type, and current strength. By studying the distribution of fossils in different sedimentary contexts, scientists can reconstruct ancient habitats and understand how environmental factors shaped the ecology of these early seas. Ediacaran ecosystems were not isolated systems, but were part of larger biogeochemical cycles. The feeding, movement, and decomposition of organisms contributed to the cycling of nutrients such as carbon, nitrogen, and phosphorus. The breakdown of organic matter, whether by microbial decomposition or absorption by organisms, released nutrients back into the environment, supporting further microbial growth and sustaining the food web. This interplay between organisms and chemical cycles illustrates that even in these ancient seas, life was already deeply intertwined with the chemistry and physics of the Earth system. While many Ediacaran organisms relied on passive feeding, the period also saw the emergence of innovative forms that expanded ecological possibilities. Some species developed branching or quilted structures that increased surface area, enhancing nutrient absorption. Others exhibited modular growth, allowing parts of the organism to continue functioning even if other parts were damaged or buried. These adaptations reflect an early experimentation with form and function, demonstrating that ecological roles were not fixed but could evolve in response to environmental pressures. The fossil record also hints at early forms of competition. Space on microbial mats was a limited resource, and organisms that could grow quickly, spread across surfaces, or occupy elevated positions likely had a selective advantage. Competition may have driven variations in body size, shape, and growth patterns, contributing to the diversity of forms observed in Ediacaran assemblages. Unlike later periods dominated by predation, competition in the Ediacaran appears to have been primarily about space and access to nutrients rather than defense against being eaten. Symbiotic relationships may also have existed. While direct evidence is scarce, it is plausible that some Ediacaran organisms engaged in mutually beneficial interactions, such as sheltering small microbial colonies or facilitating nutrient exchange. These early partnerships would have added another layer of ecological complexity, further emphasizing that Ediacaran ecosystems were dynamic and interconnected. The study of Ediacaran ecological roles provides insights not only into the lives of these ancient organisms, but also into the broader trajectory of life on Earth. By examining how these early multicellular forms fed, moved, reproduced, and interacted with their environment, scientists can trace the origins of fundamental ecological processes that continue to operate in modern ecosystems. Energy flow, nutrient cycling, habitat modification, and community structure, all essential components of contemporary ecology, were already emerging during the Ediacaran, showing that even in the earliest complex seas, life was actively shaping the world around it. In summary, the Ediacaran period represents a crucial stage in the evolution of life on Earth, where complex multicellular organisms began to establish themselves within ecological networks. These early animals and frond-like forms played diverse roles, from passive feeders absorbing nutrients from microbial mats to slow-moving grazers like Dickinsonia. They shaped the sedimentary environment, influenced microbial communities, and experimented with a wide range of body plans and reproductive strategies. Although predation was rare, competition for space and resources and potential symbiotic interactions added layers of ecological complexity. The fossil record reveals not a static or primitive ecosystem, but a vibrant, interconnected world where life had begun to harness its environment, setting the stage for the dramatic evolutionary innovations of the Cambrian period that followed. By studying these ancient ecological roles, we gain a deeper understanding of the origins of biodiversity, the evolution of ecosystem dynamics, and the profound ways in which life and environment have always been entwined. The interactions between Ediacaran organisms and their environment, including their feeding strategies, movement patterns, reproductive methods, and relationships with microbial mats, demonstrate that even in a world without predators, life was already sophisticated, adaptable, and ecologically innovative. These first complex ecosystems were laboratories of evolution, experimenting with forms, functions, and behaviors that would resonate throughout the history of life. The Ediacaran seas, once thought to be simple or monotonous, were in fact arenas of ecological creativity, providing the foundational patterns for the intricate web of life that continues to flourish on Earth today. The Ediacaran seas may seem distant and alien, yet the ecological principles that govern them are familiar, echoing in the shallow coastal waters, tidal flats, and microbial-rich sediments of today's oceans. This remarkable period of life set the stage for the evolutionary explosion that would follow. The Ediacaran organisms, with their diverse shapes, feeding strategies, reproductive methods, and interactions with the environment, laid the groundwork for the ecological complexity that defines life on our planet. The Ediacaran period, stretching roughly from 635 million years ago to 540 million years ago, represents one of the most mysterious and fascinating chapters in the history of life on our planet. During this time, the Earth's oceans were home to a remarkable array of soft-bodied organisms whose forms, structures, and ecological roles were unlike anything seen in later periods. Among the many mysteries surrounding these early life forms, one of the most intriguing questions is how they reproduced and what their life cycles might have looked like. Unlike modern animals, which often leave behind bones or shells, Ediacaran organisms mostly consisted of soft tissues, which rarely fossilized. As a result, scientists must rely on indirect evidence, fossil impressions, patterns in sediment, and comparisons to living organisms to form hypotheses about reproduction in these ancient beings. Even with these challenges, researchers have developed several fascinating theories that provide glimpses into how life may have continued and spread in these ancient seas. One of the first points to consider when examining reproduction in the Ediacaran biota is that these organisms were likely very different from the animals we are familiar with today. Many of them lacked clear body parts such as mouths, guts, or limbs. This has led scientists to explore the idea that reproduction might have been simpler and more flexible, similar in some ways to certain modern sea creatures such as sponges, corals, and some flatworms. One prominent hypothesis is that many Ediacaran organisms reproduced asexually, meaning that a single individual could produce offspring without the need for a partner. Asexual reproduction is an efficient way to increase population numbers quickly, especially in a relatively stable and uncompetitive environment like the shallow seas of the Ediacaran. There are several forms of asexual reproduction that scientists have considered for these organisms, each providing different possibilities for the life cycles of these early animals. Fragmentation is one form of asexual reproduction that could have been common in the Ediacaran. In fragmentation, a portion of the organism breaks off, and that piece grows into a complete new individual. Fossil evidence suggests that some Ediacaran species, such as Dickinsonia, which had a flat, segmented body, may have been capable of this process. Researchers have observed repeated patterns in fossil specimens, where smaller individuals appear to resemble parts of larger ones, hinting that parts might have separated and developed independently. This method of reproduction would allow a population to spread over large areas of the seafloor without the need for complex reproductive organs or behaviors. Fragmentation also has the advantage of producing clones, meaning offspring are genetically identical to the parent, which could be beneficial if the parent was well adapted to its environment. Another type of asexual reproduction that could have occurred in the Ediacaran is budding, a process still seen today in some cnidarians like corals and hydras. In budding, a new individual grows out of the body of the parent organism and eventually detaches, becoming independent. Some frond-like organisms, such as Charnia, may have used budding to expand their populations. Fossil evidence shows clusters of similarly sized individuals arranged in ways that suggest new organisms could have sprouted from the parent's body, forming dense colonies. This method would not only increase numbers, but also help organisms anchor themselves to the substrate, making it easier to remain in nutrient-rich areas and survive in the dynamic shallow seas. While asexual reproduction seems to have been common, many scientists also believe that sexual reproduction may have been present in the Ediacaran, even if it was not universal. Sexual reproduction, which involves the fusion of genetic material from two individuals, introduces variation and allows populations to adapt more quickly to changing environments. The main challenge is that direct evidence of sexual organs or gametes has not been found in Ediacaran fossils, largely due to their soft-bodied nature. However, some researchers suggest that certain forms of Ediacaran life, especially those with bilateral symmetry, like Sprigina, may have had primitive sexual reproduction. These organisms may have released eggs and sperm into the surrounding water, where fertilization occurred externally. This is a method still used by many modern marine invertebrates, such as sea urchins and some worms, and it could have been an early strategy for increasing genetic diversity in ancient oceans. The life cycles of Ediacaran organisms would likely have been influenced by environmental conditions, including water temperature, nutrient availability, and oxygen levels. During the Ediacaran, oxygen levels in the oceans were rising, but were still lower than what we see today. Many organisms may have relied on simple diffusion to exchange gases, which could limit their size and influence how they reproduced. For instance, smaller organisms that reproduced asexually could spread quickly across the seafloor without needing complex circulatory systems. Larger frond-like forms may have developed reproductive structures at specific times of the year, or in response to seasonal nutrient pulses, much like modern marine plants and corals that synchronize spawning events to maximize survival of offspring. Some scientists have proposed that the Ediacaran organisms' life cycles may have included multiple stages, including larval forms. Larval stages are common in many modern marine animals, providing a way for young individuals to disperse before settling into a fixed location. Although direct larval fossils are rare, some small-scale fossil patterns suggest that tiny juvenile forms may have existed and gradually grew into larger adults. This would allow populations to colonize new areas, maintain genetic diversity, and avoid competition for resources among adults. In the case of frond-like organisms, young individuals may have floated or drifted in the water column before anchoring themselves to the substrate and developing the full adult structure. Such a life cycle would represent an important adaptation to the dynamic and sometimes harsh conditions of shallow Ediacaran seas. Microbial mats, which were widespread on the seafloor during the Ediacaran, may have also played a critical role in reproduction and life cycles. These mats provided not only food, but also a surface for attachment and growth. Some researchers suggest that the close association of Ediacaran organisms with microbial mats may have facilitated asexual reproduction through budding or fragmentation. For example, as an organism grew, it could produce new individuals directly on the mat, creating clusters of interconnected forms. The microbial mats themselves could help protect young individuals from being swept away by currents and may have provided chemical signals that triggered reproductive events. This intimate relationship between organisms and microbial mats highlights the interconnectedness of Ediacaran life and suggests that reproduction was not only a biological process, but also an ecological one, influenced by the surrounding environment. Reproduction in the Ediacaran may also have involved seasonal or environmental cues, similar to many modern marine species. Changes in water temperature, oxygen concentration, or nutrient availability could have signaled optimal times for reproduction, whether through asexual or sexual means. Fossil evidence shows certain species appearing in clusters or layers, hinting at synchronized reproductive events that allowed populations to expand rapidly during favorable conditions. This type of reproductive strategy would have been advantageous in the often unpredictable shallow seas of the time, where sudden changes in sediment deposition or water currents could dramatically alter the landscape. Some Ediacaran organisms, especially those with radial or fractal symmetry, such as fractal-like rangeomorphs, may have had a very different approach to reproduction. These organisms appear to have grown by repeating modular units, almost like a branching tree. Each unit could potentially become independent or continue growing as part of the whole, blurring the line between growth and reproduction. In this sense, their life cycle may have been continuous, with no clear distinction between juvenile and adult stages. Instead, these organisms could have expanded through repeated growth patterns, producing new modules that functioned as both extensions of the parent and as potential new individuals. This unique form of reproduction and growth highlights how radically different life could be in the Ediacaran compared to later periods. The debate over Ediacaran reproduction is further complicated by the scarcity of complete fossils and the difficulty of interpreting soft-bodied forms. Many specimens are preserved as flat impressions on sandstone or as subtle traces in microbial mats, making it challenging to identify reproductive structures with certainty. Despite these limitations, researchers continue to uncover new fossil sites around the world, from the famous Ediacara Hills in Australia to Newfoundland in Canada and the White Sea region in Russia. Each new discovery provides additional clues, whether through the arrangement of individuals, the presence of small buds, or evidence of fragmentation. Over time, these findings are helping scientists build a more detailed picture of how reproduction and life cycles may have functioned during this enigmatic period. Some theories even suggest that Ediacaran organisms may have experimented with both sexual and asexual reproduction, depending on environmental conditions or life stage. This mixed strategy is seen in many modern organisms, such as certain jellyfish and corals, which can reproduce asexually when conditions are stable and switch to sexual reproduction when diversity is needed to survive changing environments. Such flexibility would have been a valuable advantage in the ancient oceans, allowing populations to persist through periods of nutrient scarcity, sediment disturbances, or shifts in oxygen levels. Around 635 million years ago, Earth was entering a period of remarkable transformation that would set the stage for one of the most dramatic bursts of life in its history, the Cambrian explosion. The continents were beginning to assemble into a supercontinent called Pannotia, creating shallow seas that spread across vast areas, providing new habitats for life to explore. The climate, after enduring extreme glaciations known as snowball Earth events, was slowly stabilizing. These intense ice ages had covered much of the planet, possibly even reaching the equator, and their retreat transformed the oceans and the land, creating conditions that encouraged life to experiment with new forms. Oxygen levels in the oceans had gradually risen over millions of years, reaching a point that allowed organisms to support more complex metabolisms. Higher oxygen concentrations made it possible for creatures to grow larger, develop specialized tissues, and move with greater energy and control. Life was no longer constrained to simple, small forms, drifting passively in the oceans. Organisms were beginning to explore new strategies for survival, predation, and reproduction. This increase in oxygen, combined with the warming climate and expanding shallow seas, created a perfect setting for evolutionary innovation. The late Ediacaran seas were home to mysterious, soft-bodied organisms that are now collectively called the Ediacaran biota. They included quilted, oval-shaped forms like Dickinsonia, tall frond-like structures such as Charnia, and segmented bilaterally symmetrical creatures like Sprigina. Many of these organisms lacked hard skeletons, which made their fossil records sparse and enigmatic, but the shapes and patterns preserved in ancient sediments reveal a rich diversity of life experimenting with new body plans. These creatures largely fed by absorbing nutrients from microbial mats on the seafloor or through other passive methods, and their interactions with the environment were subtle but significant, altering sediment structures and creating microhabitats that would later support more complex life forms. At the same time, geological processes were reshaping the oceans and continents. Erosion of the newly exposed land brought minerals and nutrients into the seas, fertilizing microbial communities and supporting larger ecosystems. The chemical composition of seawater, including levels of calcium, phosphate, and silica, gradually changed in ways that favored the development of hard body parts. These changes would later enable the evolution of shells, exoskeletons, and other mineralized structures, a hallmark of the Cambrian explosion. The rise of predation, a new biological pressure, also played a crucial role in this transition. By the very end of the Ediacaran period, some evidence suggests that early predators may have begun to appear, targeting soft-bodied organisms. This created an evolutionary arms race. Prey species needed defenses, whether through mobility, protective coverings, or chemical deterrence, while predators evolved better strategies to capture and consume them. This dynamic interaction set the stage for rapid diversification of body forms and ecological roles in the Cambrian seas. The emergence of new ecological niches further encouraged evolutionary experimentation. Shallow marine environments offered a variety of habitats, from calm lagoons to turbulent tidal zones, each presenting unique challenges and opportunities for survival. Burrowing behavior began to appear, allowing animals to exploit sediment layers for food or shelter. This not only changed the behavior of individual organisms, but also transformed the seafloor itself, aerating sediments and promoting new microbial and animal interactions. These activities created feedback loops, where biological innovations altered the environment, which in turn opened up further possibilities for evolution. At the molecular and developmental levels, changes were occurring that would profoundly influence the trajectory of life. The evolution of more complex genetic regulatory networks allowed organisms to control the growth and patterning of tissues in new ways. This made it possible to produce repeated structures, symmetry, and segmentation, features that would become prominent in the Cambrian fauna. Developmental flexibility meant that small genetic changes could produce large differences in body form, accelerating the pace of evolutionary experimentation. By the beginning of the Cambrian period, roughly 540 million years ago, the stage was set for an unprecedented diversification of life. The combination of rising oxygen levels, nutrient-rich oceans, new ecological pressures such as predation, and the appearance of developmental innovations created conditions that allowed life to explode into a vast array of forms. Organisms began to develop hard shells, spines, and other structures that improved mobility, defense, and feeding. These innovations, in turn, enabled the establishment of complex food webs, with primary producers, herbivores, predators, and scavengers interacting in ways that had never existed before. The transition from the late Ediacaran to the early Paleozoic was therefore not a single moment, but a prolonged period of environmental and biological experimentation. It was a time when the Earth's atmosphere, oceans, and continents converged to create opportunities for life to grow more complex. It was also a time when evolutionary pressures favored new forms, strategies, and interactions, setting the groundwork for the Cambrian explosion, which would soon fill the oceans with a dazzling variety of animals, from trilobites and brachiopods to the first primitive echinoderms and mollusks. This period demonstrates how deeply interconnected environmental changes and biological evolution are. Rising oxygen levels enabled larger, more active organisms. Shifting climates and retreating glaciers created new habitats. Erosion and mineral influx enriched the oceans. Early predation drove the evolution of defense and mobility. Developmental innovations provided the tools for creating complex body structures. Together, these factors transformed Earth from a world dominated by soft, simple organisms into a dynamic and complex ecosystem teeming with new forms of life, laying the foundation for the incredible diversity of the Paleozoic era. This remarkable transformation from the Ediacaran to the Paleozoic, spanning roughly 100 million years, is evident in the fossil record as it shifts from enigmatic soft-bodied organisms to a wealth of mineralized forms, increasingly sophisticated ecological interactions, and the appearance of modern phyla. These changes illustrate the profound impact of environmental pressures on evolution and highlight the intricate relationship between Earth's physical processes and the biological innovations they support. By studying this transition, scientists gain insight not only into the origin of complex life, but also into the processes that drive evolution on a planetary scale, showing how life can respond to new opportunities and challenges over immense spans of time. This explanation demonstrates the gradual accumulation of conditions, climatic, chemical, ecological, and genetic, that together led to the Cambrian explosion. It also emphasizes the continuous interplay between Earth's environment and the living organisms it supports. Every increase in oxygen, every nutrient-rich current, every new predator and prey relationship, every developmental innovation, and every shallow bay or reef system contributed to the dramatic diversification of life forms that characterize the early Paleozoic seas. The transition was a mosaic of interlinked processes, each creating ripples that amplified evolutionary potential, culminating in a period of unprecedented biological creativity and diversity. The shift from the Ediacaran period to the Paleozoic era, therefore, marks one of the most crucial turning points in the history of life. It was a period in which the planet moved from relative simplicity to extraordinary complexity, setting the stage for all subsequent animal life. By the time the Cambrian explosion fully unfolded, Earth had already become a dynamic and fertile environment capable of supporting a wide range of life strategies, from sedentary filter feeders to mobile predators, from simple multicellular organisms to increasingly complex ecosystems. This transition underlines the intricate dance between life and environment, demonstrating that major evolutionary leaps often require a convergence of multiple factors: geological, chemical, ecological, and biological, all working together across millions of years. The early Paleozoic era, beginning roughly 540 million years ago and continuing until around 490 million years ago, was a time of extraordinary change in the life of Earth's oceans. During this period, the seas were filled with an astonishing variety of creatures, most of which were entirely new to the world. Among the most remarkable of these early marine animals were trilobites, brachiopods, and a diverse range of other organisms with hard shells. These creatures not only shaped the ecosystems of the early Paleozoic seas, but also left behind some of the most detailed and fascinating fossils that scientists use today to understand life hundreds of millions of years ago. To appreciate the richness of this period, it is important to explore who these creatures were, how they lived, and the ways they adapted to their watery world, as well as the changes in the environment that allowed them to thrive. Trilobites were among the most iconic animals of the early Paleozoic. These creatures, which belonged to a group of arthropods, the same larger group that includes modern insects, spiders, and crabs, were remarkable for their segmented bodies and hard exoskeletons. Their bodies were typically divided into three main parts: a head, called a cephalon, a central body called a thorax, and a tail section known as a pygidium. The name trilobite itself means three lobes, referring to the longitudinal ridges that ran along their bodies, separating the left, central, and right sections. Trilobites were incredibly diverse, with thousands of species emerging during the early Paleozoic. Some were small, measuring less than a centimeter long, while others could grow to lengths of over 70 centimeters, although such giants were rare. Their hard exoskeletons were made of a mineral called calcite, which helped protect them from predators and also allowed their bodies to be preserved as fossils over hundreds of millions of years. This preservation is why trilobites are so well known to paleontologists today. Fossil trilobites have been found on every continent, showing just how widespread they were in Paleozoic oceans. Trilobites were not all alike in how they lived. Some species were active swimmers, moving through the water to hunt for small prey, while others scuttled along the sea floor, searching for organic particles or small organisms to eat. Many trilobites had large, compound eyes, which were made up of hundreds of tiny lenses, giving them an extraordinary ability to detect movement and light changes in their surroundings. This would have helped them avoid predators and find food in the dim light of the ocean depths. Trilobites also molted their exoskeletons, shedding their old shells as they grew, much like modern crabs and lobsters. These molted exoskeletons are often found in fossil beds alongside fully grown trilobite skeletons, giving scientists insight into how these creatures grew and developed over their lifetimes. Alongside trilobites, brachiopods were another dominant group in early Paleozoic seas. Brachiopods are marine animals with two hard shells, or valves, that cover the upper and lower parts of their bodies. They may resemble clams at first glance, but brachiopods are a completely different group, with a very different internal structure. Brachiopods attach themselves to the seafloor using a stalk-like structure called a pedicle, which anchored them securely in place, allowing them to filter tiny particles of food from the surrounding water. They were not mobile like many trilobites, but their stationary lifestyle was highly successful in the shallow seas of the time. During the early Paleozoic, brachiopods were especially abundant, and they came in a wide range of shapes and sizes, from small, delicate shells to larger, more robust forms. Their shells were made of calcium carbonate, the same mineral found in modern seashells, which contributed to their excellent fossil preservation. The diversity of brachiopods in the early Paleozoic was extraordinary. Different species occupied different ecological niches, from shallow tidal flats to deeper, calmer waters. Some had smooth shells, while others had ridged or spiny surfaces, which may have helped them blend into their environment or deter predators. Brachiopods reproduced by releasing eggs and sperm into the water, allowing fertilization to occur externally, which meant their young developed as free-swimming larvae before settling on the sea floor to grow into adults. This method of reproduction allowed brachiopods to colonize wide areas of the oceans and maintain their presence throughout the early Paleozoic. Besides trilobites and brachiopods, the early Paleozoic seas hosted many other shelled organisms that played important roles in the ecosystems of the time. For instance, mollusks, which include early relatives of snails, clams, and cephalopods, became increasingly diverse during this era. Primitive mollusks were often small and simple, but by the later stages of the early Paleozoic, more complex forms like early cephalopods began to appear. These cephalopods, related to modern squids and octopuses, had chambered shells that allowed them to control their buoyancy and move through the water with surprising agility. Other important shelled animals included hyoliths, which were cone-shaped organisms that lived on the sea floor, and various echinoderms, such as early relatives of starfish and sea urchins, which had rigid skeletons made of calcium carbonate plates. The proliferation of shelled organisms during the early Paleozoic was closely tied to changes in the chemistry of the oceans. Around 540 million years ago, the oceans had higher concentrations of calcium and carbonate ions, which made it easier for animals to produce hard skeletons and shells. This chemical shift likely contributed to what scientists call the Cambrian explosion, a rapid increase in the diversity of life forms that had begun in the preceding Ediacaran period and continued into the early Paleozoic. Hard shells provided numerous advantages. They offered protection from predators, structural support for larger bodies, and new opportunities for movement and feeding strategies. The development of shells also influenced the evolution of predators, which in turn drove further innovations in defensive adaptations among shelled organisms. Predation became an important ecological force during the early Paleozoic. Trilobites, brachiopods, and mollusks were all subject to attacks from early predators, such as large arthropods or primitive cephalopods. Some trilobites developed spines or thickened exoskeletons to deter attackers, while brachiopods evolved more complex shell shapes and thicker shells for protection. Evidence of predation is preserved in the fossil record, often in the form of bite marks or repaired shells, showing that interactions between predators and prey were shaping the course of evolution. These interactions led to a biological arms race, where adaptations and counter-adaptations spurred rapid diversification and contributed to the complex ecosystems of the early Paleozoic. The arrangement of early Paleozoic seas also played a crucial role in shaping marine life. Continents were positioned differently than today, with most land masses located near the equator. Shallow continental shelves and warm, sunlit waters created ideal habitats for benthic communities, animals living on or near the sea floor. These shallow marine environments were rich in nutrients, supporting dense populations of trilobites, brachiopods, and other invertebrates. In deeper waters, fewer organisms thrived, but the few that did were often highly specialized. The distribution of species across these different marine habitats contributed to the remarkable diversity observed in the fossil record. Fossil evidence from early Paleozoic rocks shows intricate communities of shelled animals living together in complex ways. In many fossil beds, trilobites are found alongside brachiopods, mollusks, and other invertebrates, suggesting that these species coexisted in large, diverse communities. Some trilobites may have scavenged organic matter left by other animals, while brachiopods filtered microscopic particles from the water. This combination of mobile and stationary lifestyles, predators and prey, contributed to a dynamic and interconnected ecosystem. Fossilized burrows and trackways also reveal that many trilobites actively explored the seafloor, leaving behind traces of their movement that provide insights into their behavior. In addition to their ecological roles, the early Paleozoic shelled organisms had a profound impact on the geological record. As these animals died, their calcium carbonate shells accumulated on the seafloor, eventually forming thick layers of limestone and other sedimentary rocks. These fossil-rich deposits preserve a vivid record of the biodiversity of the time and serve as a window into the ancient oceans. Studying these rocks allows scientists to reconstruct ancient ecosystems, understand environmental conditions, and trace the evolution of life over millions of years. In particular, trilobite fossils are used by geologists as index fossils, helping to date rock layers and correlate them across different continents. The early Paleozoic was also a time of significant evolutionary experimentation. New body plans, feeding strategies, and defensive adaptations emerged as shelled animals diversified. Trilobites evolved a wide range of forms, from flattened burrowers to spiny predators. Brachiopods experimented with shell shapes and internal structures, while mollusks began exploring new ways of swimming, crawling, and feeding. This period set the stage for further evolutionary milestones in the later Paleozoic, including the rise of reef-building organisms and more complex food webs. Environmental factors also influenced the distribution and evolution of early Paleozoic marine life. Changes in sea level, climate fluctuations, and shifts in ocean chemistry created new habitats and opportunities for colonization. Warm, shallow seas promoted the spread of brachiopods and trilobites, while cooler, deeper waters supported specialized communities. Periodic events such as transgressions, when sea levels rose and flooded continental areas, and regressions when sea levels fell, reshaped the available habitats and drove local extinctions and migrations. These environmental dynamics, combined with biological innovations, fueled the remarkable diversification of marine life during this era. It is also worth noting that early Paleozoic marine ecosystems were not dominated solely by shelled animals. Soft-bodied organisms, including worms, sponges, and early relatives of corals, were important components of these ancient seas. Though less likely to fossilize, these creatures contributed to the ecological complexity of the time. Sponges filtered water, worms burrowed into sediments, and primitive coral-like organisms began forming small reef-like structures. Together with trilobites, brachiopods, and mollusks, these soft-bodied animals created rich and intricate marine ecosystems that supported a wide range of life forms. The legacy of early Paleozoic shelled organisms continues to shape our understanding of life on Earth. Fossils of trilobites, brachiopods, mollusks, and other invertebrates provide crucial evidence of how life responded to environmental changes, how ecosystems evolved, and how new body plans emerged. These early animals also demonstrate the importance of hard parts in evolution, showing how the development of shells and exoskeletons opened new ecological possibilities. By studying these ancient creatures, scientists can trace the roots of modern marine ecosystems and gain insights into the long history of biodiversity on our planet. Paleozoic plant life marked one of the most profound transformations in Earth's history, shaping landscapes, climates, and ecosystems in ways that would endure for hundreds of millions of years. During this time, from roughly 545 million years ago to about 252 million years ago, plants underwent remarkable innovations that allowed life to move from water-dominated environments onto dry land. These changes were not sudden. They unfolded gradually over tens of millions of years, beginning with the simplest non-vascular plants and advancing to more complex vascular forms, which could transport water and nutrients throughout their structures. This evolution of plant life had far-reaching consequences, altering soils, the atmosphere, and the very structure of ecosystems on Earth. The earliest land plants were non-vascular, small, and fragile, similar in appearance to modern mosses and liverworts. They lacked specialized tissues to carry water or nutrients internally, relying instead on direct absorption from the surrounding environment. This limitation confined them to moist areas near lakes, rivers, and coastal wetlands. Despite their simplicity, they played an essential role in modifying their environment. By covering rock surfaces and interacting with the mineral-rich substrates, they slowly contributed to soil formation. This process not only provided a medium for future plant life, but also initiated a long-term transformation of landscapes. Over time, the accumulation of organic material from decaying plant matter enriched these soils, creating conditions that could support larger and more complex plants. The evolution of vascular tissue marked a major turning point in Paleozoic plant life. Xylem and phloem allowed plants to transport water, minerals, and sugars efficiently, giving them the capacity to grow taller and colonize areas farther from water sources. The earliest vascular plants appeared during the late Silurian period, around 430 to 425 million years ago. These plants, such as Cooksonia, were small, leafless, and characterized by simple branching stems with spore-producing structures. Their appearance opened the door to the first significant colonization of terrestrial habitats. Unlike their non-vascular predecessors, these vascular pioneers could stabilize larger areas of soil, influence erosion patterns, and contribute more significantly to the cycling of nutrients in early terrestrial ecosystems. Reproduction in early vascular plants relied on spores rather than seeds, a feature inherited from their aquatic ancestors. These spores could be carried by wind or water to new locations, promoting the spread of plants across increasingly vast regions. Over time, adaptations such as protective layers around spores and structural reinforcements in stems allowed plants to withstand drier conditions and colonize environments that were previously inhospitable. These innovations contributed to the gradual formation of the first plant-dominated landscapes, laying the groundwork for the emergence of dense vegetation during the Devonian period, approximately 420 to 360 million years ago. During the Devonian, plant life became more diverse and structurally complex. Leafless stems gradually gave rise to true leaves, increasing the surface area for photosynthesis and allowing plants to capture sunlight more efficiently. The development of roots enabled them to anchor more securely in the soil, absorb water from greater depths, and extract essential nutrients. These changes had profound ecological consequences. Plant roots penetrated and fractured rock, accelerating soil formation and altering drainage patterns. The accumulation of plant biomass increased organic matter in soils, enhancing their fertility and stability. As plants grew taller and more abundant, they provided habitats for the earliest terrestrial animals, such as arthropods and early tetrapods, which were beginning to explore life on land. The Devonian also saw the emergence of the first true forests, a milestone in the history of terrestrial life. These early forests were composed of primitive trees like Archaeopteris, which combined fern-like leaves with woody stems. Spanning tens of meters in height, these trees transformed landscapes, creating shaded environments, influencing local climate conditions, and contributing to the first complex terrestrial ecosystems. Forests slowed runoff, reduced soil erosion, and promoted the development of intricate food webs. Decaying plant matter enriched the soil and formed the basis for the first widespread terrestrial carbon deposits, which later influenced the formation of coal during the Carboniferous period. By the Carboniferous, roughly 359 to 299 million years ago, plant life reached an unprecedented level of complexity and abundance. Vascular plants had diversified into ferns, horsetails, club mosses, and seed-producing plants. Swampy regions, particularly near equatorial zones, were dominated by towering lycophytes such as Lepidodendron, which could reach heights of up to 40 meters. These massive plants had thick, scaly trunks and extensive root systems that stabilized wetland soils and created ideal habitats for a wide variety of organisms. Alongside lycophytes, tree ferns and seed ferns expanded across floodplains, contributing to dense vegetation that regulated water cycles and provided abundant organic matter for emerging terrestrial food chains. One of the most important ecological contributions of Paleozoic plants was their effect on the atmosphere. Through photosynthesis, Plants absorbed carbon dioxide and released oxygen, gradually increasing atmospheric oxygen levels over millions of years. This shift enabled the rise of larger and more active terrestrial animals, from insects to early amphibians. By the late Carboniferous, oxygen concentrations may have reached levels higher than today, facilitating the evolution of giant arthropods, such as dragonfly-like insects with wingspans exceeding 75 centimeters, and creating conditions that supported more complex terrestrial ecosystems. The interaction between plants and the atmosphere illustrates the profound influence of vegetation on Earth's climate and biological evolution. The expansion of seed-producing plants, or gymnosperms, began toward the end of the Paleozoic, particularly during the late Carboniferous and early Permian, around 300 million years ago. Seeds allowed plants to reproduce without relying on water for fertilization, giving them a significant advantage in drier environments. These plants included early conifers and chordates, which could thrive in upland areas and spread beyond swampy lowlands. Seed plants had structures to protect embryos, supply them with nutrients, and facilitate dispersal, enabling colonization of more diverse habitats. This adaptation not only diversified terrestrial vegetation, but also prepared the stage for the dominant forests of the Mesozoic era. Soil development continued to evolve alongside plant life. Early vascular plants had already begun altering the chemical composition of soils by excreting organic acids that broke down rocks. As plant communities became more complex, root systems penetrated deeper, mixing organic matter with minerals and enhancing nutrient availability. By the late Paleozoic, soils were rich and capable of sustaining diverse plant assemblages, which in turn supported more complex terrestrial animal life. The feedback loop between plants, soils, and animals created dynamic ecosystems in which each component influenced the others, establishing the ecological foundations that persist in modern forests and grasslands. Water regulation was another vital contribution of Paleozoic plants. By covering large portions of the land, plants slowed surface runoff, increased water infiltration, and stabilized stream banks. Dense vegetation along rivers and floodplains prevented soil erosion, reduced sedimentation in waterways, and influenced the formation of meandering rivers and wetlands. This hydrological impact shaped landscapes over millions of years, demonstrating how the presence of plants could transform the physical environment as well as the biological one. Wetland forests, in particular, created conditions favorable for the preservation of organic material, eventually leading to the formation of coal seams that remain important energy resources today. Paleozoic plants also influenced evolutionary trajectories of other organisms. The presence of abundant vegetation created new niches for insects, amphibians, and early reptiles. Plant surfaces provided food, shelter, and breeding grounds, while the complexity of forested habitats promoted diversification and specialization among animals. Insects, in particular, began forming relationships with plants, including herbivory and pollination, although true flowering plants would not appear until the Mesozoic era. The rise of vascular plants, forests, and seed plants during the Paleozoic thus directly contributed to the complexity and diversity of terrestrial ecosystems. Fossil evidence provides remarkable insight into the structure, diversity, and ecological impact of Paleozoic plant life. Fossils of early vascular plants such as Cooksonia and Rhynia reveal the simple branching patterns and spore-bearing structures of these pioneers. Later fossils, including Archaeopteris and Lepidodendron, show the development of leaves, wood, roots, and reproductive structures that allowed plants to dominate landscapes. Coal deposits from the Carboniferous and Permian, formed from dense swamps of lycophytes, ferns, and seed ferns, preserve impressions of leaves, stems, and roots, providing a detailed record of plant communities and their ecological roles. These fossils not only demonstrate the physical characteristics of ancient plants, but also allow reconstruction of Paleozoic ecosystems, showing how vegetation shaped habitats and interacted with other forms of life. The development of simple vascular plants during the Paleozoic era thus represents one of the most transformative periods in Earth's history. From small, fragile, moisture-dependent pioneers to towering swamp forests and seed-producing gymnosperms, plants transformed the land, atmosphere, soils, and ecosystems. They regulated water cycles, stabilized soils, enriched the atmosphere with oxygen, and provided the foundation for complex food webs. Their evolution set the stage for the explosive diversification of life on land and the rise of the forests and deserts that would dominate subsequent eras. By the end of the Paleozoic, around 252 million years ago, plants had established themselves as fundamental architects of terrestrial life, creating conditions for the extraordinary biological diversity that continues to flourish on Earth today. The Paleozoic era demonstrates the profound influence that seemingly simple organisms can have on the planet. Simple vascular plants were not merely passive passengers in Earth's history, they were active engineers of ecosystems. Through the slow but persistent accumulation of organic matter, the expansion of root networks, and the colonization of new habitats, these plants reshaped landscapes, moderated climates, and facilitated the evolution of complex life forms. Their legacy is evident in every forest, grassland, and wetland that exists today, as well as in the fossilized remains that preserve their story across hundreds of millions of years. The development of simple vascular plants was a pivotal moment in the history of life, transforming a barren land into a living, breathing world of green that would sustain countless generations of organisms, both ancient and modern. By the time the Paleozoic era drew to a close, plant life had evolved a range of reproductive strategies, structural innovations, and ecological interactions that allowed them to dominate terrestrial environments. Spore-bearing ferns and club mosses thrived in humid lowlands, while early seed plants colonized drier uplands, illustrating the adaptability of vascular plants. The diversification of these plant groups, combined with their ability to engineer soils, influence atmospheric composition, and provide habitats for other organisms, highlights the intricate interconnections between plant life and Earth's systems. The remarkable success of Paleozoic plants laid the foundation for the Mesozoic era, during which the rise of conifers, cycads, and eventually flowering plants would further transform the world. The Paleozoic era was a time of extraordinary biological innovation and transformation. During this vast span of nearly 300 million years, life on Earth experienced incredible diversification, with new types of organisms appearing, spreading, and sometimes vanishing entirely. Among the defining features of this era were mass extinctions, catastrophic events that reshaped life on Earth, abruptly wiping out a large portion of existing species and forcing ecosystems to reorganize. These extinctions were not just momentary losses, but transformative episodes that influenced the trajectory of evolution for hundreds of millions of years. They left behind patterns in the fossil record that help scientists understand the fragility and resilience of life under changing environmental conditions. Two of the most significant mass extinctions of the Paleozoic occurred during the late Ordovician, around 445 million years ago, and the late Devonian, roughly 375 million years ago. These events profoundly affected the oceans and, indirectly, the early colonization of land by plants and animals, setting the stage for future evolutionary radiations. The late Ordovician extinction, occurring approximately 445 million years ago, is recognized as one of the earliest major extinction events in Earth's history. At this time, life was predominantly marine. The seas were rich with a variety of organisms, including trilobites, brachiopods, bryozoans, graptolites, and primitive mollusks, many of which had developed complex ecological relationships over millions of years. Coral-like organisms and stromatoporoids contributed to reef building, creating intricate habitats that supported a remarkable diversity of life. However, the stability of these ecosystems was severely challenged by dramatic changes in the Earth's climate and geography. Evidence suggests that this extinction was closely linked to a period of global cooling, during which glaciers formed and sea levels dropped significantly. These glaciations were concentrated in the southern supercontinent of Gondwana, which, at the time, was situated near the South Pole. As ice sheets expanded, shallow marine habitats, where most life thrived, were reduced, leaving many organisms without suitable living spaces. The late Ordovician extinction unfolded in two main pulses. The first pulse appears to have been driven by the onset of glaciation, which caused a rapid drop in sea levels. As oceans receded, organisms adapted to shallow waters, such as brachiopods and trilobites, experienced massive die-offs. The second pulse occurred as glaciers melted and sea levels rose again, causing abrupt changes in water temperature and chemistry that stressed marine ecosystems further. Oxygen levels in the oceans may have fluctuated, creating zones where survival was difficult. Overall, this event is estimated to have eliminated up to 85% of marine species, making it one of the most severe extinctions in the Paleozoic. Notably, organisms with narrow ecological ranges or specialized habitats suffered the most, while more adaptable or widely distributed species were better able to survive. The causes of the late Ordovician extinction are still studied and debated, but climate change appears to have been the primary driver. The movement of continents, particularly the positioning of Gondwana, influenced ocean currents and atmospheric patterns, contributing to glaciation. Changes in carbon dioxide levels and other greenhouse gases may have played a role in shifting global temperatures. Additionally, the glaciation would have disrupted nutrient cycles in the oceans, affecting primary producers like plankton, which formed the base of the food chain. As plankton populations collapsed, the effects rippled upward, causing cascading extinctions among higher trophic levels. Fossil evidence, including widespread deposits of sediment indicative of glacial activity and sudden shifts in species composition in marine strata, supports these conclusions and provides insight into how life responded to rapid environmental stress. The Devonian period, spanning from about 419 million to 359 million years ago, witnessed profound evolutionary changes. It is often called the Age of Fishes because of the remarkable diversification of fish, including jawless fish, armored placoderms, early cartilaginous fishes, and the first true bony fishes. On land, plants were spreading and becoming more complex, with the first forests of ferns, club mosses, and early seed plants beginning to stabilize soils and influence local climates. Invertebrates, including trilobites, corals, and brachiopods, continued to flourish in the oceans. Despite this flourishing of life, the late Devonian extinction, occurring around 375 million years ago, disrupted these ecosystems in dramatic ways. Unlike the relatively rapid pulses of the late Ordovician extinction, the Devonian extinction appears to have been a drawn-out series of events spanning several million years. It primarily affected marine life, particularly organisms living in reef environments. Reef-building organisms such as stromatoporoids and corals suffered extensive losses, leading to the collapse of entire reef ecosystems in certain regions. Trilobites, which had been highly diverse and abundant, experienced significant declines, and many brachiopods and ammonoids vanished from the fossil record. Fish, including placoderms and early bony fishes, faced substantial pressures, although some lineages survived and continued to diversify afterward. The causes of the late Devonian extinction were complex, and likely involved multiple factors interacting over long periods. One contributing factor was global cooling and associated oceanic anoxia, in which large portions of the ocean became depleted of oxygen. Anoxic conditions would have been especially lethal to organisms with high oxygen demands or limited mobility, making survival difficult. The spread of dense forests on land may have played a surprising role, as increased plant growth altered soil composition and nutrient runoff into the oceans. This influx of nutrients could have triggered algal blooms, leading to further oxygen depletion in marine environments. Volcanic activity and possible asteroid impacts have also been suggested as contributing factors, but evidence for these remains less conclusive than climate-driven explanations. The consequences of the Devonian extinction were profound, reshaping marine ecosystems and influencing evolutionary pathways. The loss of reef-building organisms created gaps in marine habitats that took millions of years to recover. Some groups, like ammonoids, would eventually diversify and fill ecological niches left vacant, while others, such as many placoderms, disappeared entirely. On land, plant evolution continued, with forests gradually becoming more extensive and complex, stabilizing environments and enabling the further colonization of terrestrial habitats by early arthropods and vertebrates. The extinction events of the Devonian underscored the interconnectedness of marine and terrestrial ecosystems and highlighted the sensitivity of life to environmental shifts. Both the late Ordovician and late Devonian extinctions illustrate broader patterns in Paleozoic life. These events were not isolated catastrophes, but were linked to long-term trends in climate, sea level, atmospheric composition, and continental arrangements. They show that even in an era of tremendous innovation and diversification, life was vulnerable to rapid or extreme environmental change. Extinctions provided opportunities for surviving organisms to adapt and radiate into vacated niches, driving evolutionary processes forward. After the late Ordovician, new trilobite lineages and brachiopod groups emerged, while after the Devonian extinction, fish and reef organisms diversified anew, laying the groundwork for the ecosystems of the Carboniferous and Permian periods. Studying these mass extinctions offers valuable insights into the nature of evolutionary pressures and the dynamics of life on a changing planet. Fossils serve as windows into past worlds, revealing patterns of survival and loss, adaptation and extinction. The late Ordovician extinction shows how rapid climate change can threaten marine-dominated ecosystems, while the Devonian extinction demonstrates the complexity of long-term environmental stressors and their cascading effects across ecosystems. Both events highlight the role of environmental thresholds, the vulnerability of specialized species, and the resilience of life through adaptation and diversification. These Paleozoic extinctions also help scientists understand modern biodiversity crises. The patterns observed in the fossil record, including selective survival, habitat collapse, and ecosystem restructuring, have parallels in contemporary ecological changes driven by human activity, climate shifts, and habitat destruction. By examining how ancient organisms responded to past crises, researchers can better predict potential outcomes for modern species and ecosystems, emphasizing the need for conservation and careful stewardship of Earth's remaining biodiversity. Ultimately, mass extinctions in the Paleozoic were not just endings, but also beginnings. They cleared the stage for new forms of life to evolve, setting in motion the dynamic processes that continue to shape Earth's biosphere. Life's history in the Paleozoic, punctuated by the late Ordovician and Devonian extinctions, demonstrates both the fragility and the tenacity of organisms, highlighting the complex interplay between environmental change and biological adaptation. Through these events, we see how the planet's history is written in the rise and fall of species, in the ebb and flow of ecosystems, and in the enduring drive of life to survive, diversify, and flourish in the face of immense challenges. The fossil record, painstakingly preserved in sedimentary rocks around the world, stands as a testament to these dramatic chapters in Earth's history, offering not only a window into the distant past, but also a mirror reflecting the potential trajectories of life on our ever-changing planet. The late Ordovician and Devonian extinctions were part of a broader series of extinction events that continued throughout the Paleozoic era, including the end Permian extinction, which would later become the most severe extinction event in Earth's history. Each event was shaped by the unique combination of geological, climatic, and biological factors present at the time. By examining these extinctions, scientists can trace the ebb and flow of biodiversity, understanding not only how life is lost, but also how it rebounds, adapts, and evolves in the aftermath. These episodes reveal the resilience of life and the complex interactions between the Earth's physical environment and the organisms that inhabit it, painting a picture of a dynamic, ever-changing world where survival requires constant adaptation and innovation. By the end of the Devonian, ecosystems were transformed, species compositions were altered, and evolutionary trajectories had been redirected. Marine life, though deeply affected, continued to diversify in new directions. On land, the increasing complexity of plants and the colonization of terrestrial habitats by invertebrates and vertebrates set the stage for the Carboniferous period, during which forests would expand, insects would radiate, and the first true amphibians would establish themselves as significant terrestrial vertebrates. The legacy of the Paleozoic mass extinctions is thus a story of loss and renewal, destruction and opportunity, and the enduring capacity of life to adapt and thrive even under the most challenging circumstances.